Hello and welcome. It's my pleasure to share with you today a new analysis method aimed at helping you get the most out of your high-parameter data. As our data sets become more and more complex, it becomes tricky to search through them for relevant differences, and even more difficult to know when those changes are robust. The tool I'm talking about today can help with that. Today, I'm going to talk to you about tSNE. tSNE is a data visualization tool that takes multidimensional data, such as high-parameter data from flow cytometry or single-cell RNA sequencing, and plots it in 2 dimensions. It first splats that data out randomly, and then tries to push and pull the data until the similar data points are together, and the structure is more close to the original structure in many dimensions. Now, as we see in these Google Mentions plots, tSNE popularity has exploded in the last few years. Become really quite popular. This, it's visually appealing, it's relatively quick, and it plots the data out in a way that helps us get our heads around it for an overview. The thing is that tSNE plots depend a lot on the input parameters. You take the same data, change the perplexity, the number of iterations, other elements, and you can end up with something looking quite different. As you can see here in this Nature Communications paper, they're running tSNE on the same data set with variations in the methodology. You run it twice with different random starting points, and nothing will be in the same place. This is potentially a problem for understanding how the changes are occurring and how robust they are. Once we have a tSNE, what should we do with it? Should we just look at it and say, oh, this bit over here looks a little bit different? Maybe there's something interesting going on. We can draw some dates on it the way they've done in this example here. We can use some algorithms for clustering and plot those clusters on the tSNE, as they've also done here with these colors. Does any of this actually use the tSNE coordinates, though? Wouldn't it be nice to have a way to statistically analyze the differences between the samples in our groups on the tSNE? What I'm going to show you today is that we can. We can test the distribution of the points in the tSNE and draw conclusions based on statistics about our data. I hope to convince you that these conclusions can be strong, and that there's a lot of meaning you're missing out on by not using this method. Now, before I scare anybody off with the mathematics, I'm not a mathematician, I'm an immunologist. Carlos Roca is a mathematician and invented the math statistical test that I'm going to talk about today. I'm just going to try to explain the basis for the test in a simple and hopefully correct way. So, how does tSNE work? tSNE maps the probabilities of the points in the original multidimensional space. This creates one distribution of data. It then maps the points in a new, usually 2-dimensional space, creating a second distribution in the transform space. The difference between these two is represented by this cost function, the divergence between them. So we have one map or set of coordinates, and a second map. And the difference between those two is the key. tSNE works iteratively, which is to say it repeats the same process to refine it. It starts out randomly, which is why you get a different shape every time you run it. By iterating the process and trying to minimize the cost function, tSNE slowly groups similar points together in probability space. It does this by focusing on the local similarities. Bigger distances become equivalent and are ignored. UMAP, by this way, works similarly. Carlos's insight was that we could use this Kullback-Leibler divergence on a point-by-point basis to get information about the probability distribution of the tSNE representation. The local distance differences will correspond with differences in the probability distribution, and also differences in the distribution of distances in the tSNE representation space. This difference in distributions or entropies between the original data and the tSNE representation of the data serves as the basis for the cross-entropy test, the statistical test. Why do we need to use cross-entropy? Why can't we just use the original data or the tSNE coordinates? Well, remember that tSNE distorts the data, emphasizing differences while preserving similarity. So statistical testing on the original probability space has little power because it hasn't been grouped. And testing on the t-SNE space is not correct. It's too likely to lead to low p-values, again, because it's emphasized those differences. What does this mean? Well, if we generate two t-SNE distributions of the same original data, they will look quite different due to random initialization. You can see that the colored clusters in these two examples running off of different initializations, they look about the same, but they're in different places. These are the same cells in both examples. They're rotated around and shuffled, so nothing's in the same place. Nothing has the same coordinates. But the distribution of cross entropies, or KL divergences between the original data and the t-SNE data, is the same. Here we're looking at the cumulative distribution function of the cross entropies for each plot laid on top of each other. The two lines are indistinguishable. So the test can tell that these two examples are identical. In order to thoroughly test this new statistical method, we needed some new high parameter data. So I generated some high parameter flow cytometry data using the Aurora spectral analyzer, some mouse cells, and the panel you see here. This panel covers 50 markers and autofluorescence and identifies the major immune cell types. In addition to separating basic cell types, we get some handy activation markers, which we'll use later. It's been about a year since I did this, and new products have come to market, so you should be able to make a better panel than this. Here's some examples of how the data looks like in 2 dimensions, which is how we frequently look at flow cytometry data. We can identify monocytes and neutrophils, eosinophils, macrophages, dendritic cell subsets, as well as some NK cells, ILCs, B cell, and T cells, and some phenotyping of the activation status of these cells. So now we're going to use that high parameter flow cytometry data to assess how the cross entropy test works. First test with this data is the least interesting from a biological point of view, but the most important for statistical correctness. We analyze the same data, in this case lymph node cells, repeatedly. We can generate distinct t-SNE plots with this characteristic rotational symmetry. In this ideal case, the cross entropy test gives a p-value of 0.585, so not statistically significantly different. However, we need to know whether the test will be correct more than just once. How reliable is it? And therefore, how correct is it? For that, we run the t-SNE and compare the cross entropies 1,000 times. Each time we get a p-value, as we did here, and we plot the distribution of those p-values. If the test is correct, it will randomly give a p-value of 0.05 or less exactly 5% of the time. That's what that means. Moreover, the distribution of p-values will match the uniform distribution, or a straight line. That is exactly what we see. That means that the test is neither overly nor inadequately sensitive. Now, for a more relevant case. Here we have 3 samples of spudocytes from the same mouse. They've all been stained simultaneously with that 50 color panel and plotted with t-SNE. The only variation here should be my pipetting. Visually, they look pretty similar. And the cross entropy test agrees. No p-values below 0.05. They are statistically indistinguishable. In mouse research, we generally use inbred mice to eliminate variation. In this case, I'm showing you 3 t-SNE plots, 1, 2, 3, taken from 3 different mice. The cross entropy test shows them to be indistinguishable. Yeah. So far, I've shown you a lot of examples that are pretty uninteresting, all negative results. Let's look at something else. These plots are 50 color flow data from lymph nodes, spleen, and the small intestinal lamina propria, the gut. Visually, they're all quite different, and some clusters are essentially missing in each plot. Blue is missing here. The test says that the probability that these are the same is essentially 0. Once the probability reaches a certain point, the algorithm rounds down to 0, as it is done here. In addition to simply testing whether the distributions of cross entropies are different, we can get an idea of how different they are by the magnitude of shift in the curve. This distance metric is quantitative. It can be used to compare how far apart the samples, like these different green lines, populations, like the green versus the blue, or the clusters are. This distant measure allows us to construct relationships between the groups we're looking at. For this example, I've created artificial samples by spiking in gut cells with spleen cells and vice versa. So now, in addition to the original spleen and gut tissue samples, we have a 90-10 spleen to gut sample called spleen tissue, and a 10 to 90 gut to spleen sample called tissue spleen. Visually, you can see how the green cluster fades out and the blue cluster fades in as we move from one extreme to the other. With the cross-entropy test, we can measure the difference between the distributions of each condition and construct a tree, a dendrogram. And voila, the sample that is 90% spleen is closest to full spleen, and 90% gut is closest to full gut. Oftentimes, we have results where there's a change in phenotype, but not cell frequencies, or vice versa. Does the test work in these cases? To test this, first off, I took spleen and lymph nodes, which have substantial differences in the distribution of cells. Spleen contains a lot more B cells and myeloid cells. You can see that here coming up in the differences in clusters. We also have differences in the expression of activation markers. For instance, lymph node T cells are more naive. I created artificial samples comprised solely of lymph node cells, but with the same frequency of cell types as are present in the spleen. You can see this here by comparing the green, which is the spleen, and the blue, which is the lymph node adjusted to spleen bars. Initially, the lymph node starts out with the red distribution of cells, and after the adjustment aligns with spleen. So the only difference is the expression levels, not the cell types. While the t-SNE plots are quite similar to the eye, cross-entropy test says that they are radically different due to intra-cluster differences. Conversely, we can test whether changes in cluster or cell frequency can be detected independently of activation status. For this, I generated an artificial sample using spleen cells in the same proportions as are present in the lymph nodes. I then compared them to the original spleen. While the clusters are visibly different, the expression levels are the same because the cells come from the same source. Again, the cross-entropy test determines that it is highly unlikely that these are the same. So far, I've been showing you flow cytometry data, but this works for any data you want to use for your t-SNE. Here are some examples using CyTOF, so mass cytometry, and 10x single-cell RNA sequencing on COVID patients in both cases. Using the cross-entropy test on the single-cell RNA sequencing data, we were able to reproduce the differences that the authors found in the original paper, where we're also able to identify new differences that have been overlooked by the authors. And what's more, we're able to quantify the scale of changes in each cell population using the distance metric. So that we can say that the lymph nodes, the lung neutrophils, so we can say that in the lung, neutrophils and monocytes are more affected by COVID than epithelial cells, even though the epithelial cell change is easy to identify because there's lots of cells. Maybe you prefer to use UMAP because you like the way it looks. UMAP works quite similar to tSNE, preserving local structure at the expense of distant relationships, and the cross-entropy test works on both. The test also gives correct results on SOM-based embedding. So if you use FlowSOM and like the results you get with self-organizing maps, you might consider using these square plots generated by embed SOM and using the cross-entropy test to compare them. If you'd like to use the test in your research, and we hope you do, we provided resources online. In addition to the paper and the R script, for both flow and single-cell RNA sequencing analysis, you'll find some instructional videos on our website. These are aimed at users who have little to no experience with R programming. This project was a brainchild of Carlos Roca, came up with the idea, worked out the math, and created that very useful flow cytometry analysis script based around it. He's now working as the Associate Director of Bioinformatics at AstraZeneca. Samar worked out the code for using it with single-cell RNA sequencing, and he and Julika put together the analysis of the CyTOF and 10x data. And this work would not have been possible without the support of the ERC, the Wellcome Trust, and UKRI, or the guidance of Adrian Liston. Thanks for listening.